Hello everyone and welcome back to AP Bio with AP Playground. Today we're reviewing DNA and RNA structure in topic 6.1. Objectives today are as follows. We have to describe the structures involved in passing hereditary information, including DNA and RNA molecules, as well as describing the characteristics of both of these molecules that allow them to be used as hereditary material. And our suggested skill is Concept Explanation 1C, explain biological concepts, processes, and/or models in applied contexts. First, we need to review DNA and RNA. And so DNA and RNA store and transmit genetic information, and they're nucleic acids made up of nucleotides, that is their monomer or building block. And nucleotides consist of three parts. There's going to be a 5-carbon sugar, a phosphate group, and nitrogenous base. And together, these are going to form the two strands of DNA that you see here. And so they're in the shape of a double helix. This was discovered by Rosalind Franklin. And these strands are anti-parallel, meaning they are parallel, but they run in opposite directions. And what these two strands allow for is the protection of the nitrogenous bases inside and also the storage of this genetic information. And the quote-unquote handles of this DNA ladder are formed from the 5-carbon sugar and phosphate group covalently bonding each nucleotide together, whereas the rungs themselves are formed through base pairs through hydrogen bonds between certain nitrogenous bases. And the base pairs are as following. Because we have our thymine, cytosine, and adenine, guanine nitrogenous base, then we have binding of apple, tree, car, garage, ATCG. Those are just a few ways to remember, but adenine is going to be binding to thymine, cytosine is going to be binding to guanine. These are the nitrogenous base pairs that allow the two strands to be bound together as one DNA molecule. And then the type of 5-carbon sugar we're looking at is deoxyribose, whereas RNA is going to have ribose. On the other hand, RNA only has a single strand. That makes it easier to move out of the nucleus for its functions that we'll discuss in future videos, and it's a very temporary molecule. It's going to have a ribose sugar, and because this ribose sugar contains the oxygen in the hydroxyl group, unlike the deoxyribose, which is missing an oxygen, it is now more electronegative. So base pairs will not be formed unless they are the perfect match, unless they are the base pairs that work with RNA. And RNA also has different nitrogenous bases. Instead of adenine, thymine, cytosine, and guanine, we do have cytosine. There is adenine, there is guanine, but uracil replaces thymine. So we have a replacement for T. So now our base pairs are A, U, and C, G, uracil instead of thymine. And so you can see here that these nitrogenous bases within this strand of RNA, they can bind more nucleotides to the other side of this strand. And that's going to be extremely crucial when it comes to talking about the function of RNA in transmitting genetic information and then expressing this genetic information through protein synthesis, which we'll discuss as this unit moves forward. Something else to note that you may have seen is that nitrogenous bases that form base pairs are either 1 or 2 ringed. They're either purines or pyrimidines. And so pyrimidines are the 1-ringed nitrogenous bases, and purines are the 2-ringed nitrogenous bases. And it's a little funny because one is, you would think that one is shorter, but it's the longer word. So you can see here that thymine is going to be a pyrimidine while adenine is a purine. The nitrogenous base has 1 ring, and here this one has 2. And here this one's going to have 2 rings. That one has 1 ring. And as we discussed, these nitrogenous bases are going to bind together through hydrogen bonds. However, the binding between guanine and cytosine is slightly stronger. There are 3 hydrogen bonds created, whereas between adenine and thymine there are only 2 hydrogen bonds created. And when we look at DNA as a unit, moving past the nucleotide monomer distinction, and we're looking at DNA as a nucleic acid as a polymer, these DNA strands are going to wrap around histone proteins, as you can see right over here, which form what's known as a nucleosome. And nucleosomes, in turn, are going to make up, with enough of them there, chromosomes, or chromatin, which is just condensed genetic information. And if it becomes even more condensed, then it becomes the chromosome form that we usually associate with DNA. And each chromosome is made up of introns and exons. Those are going to be coding and non-coding regions. So introns are going to be the non-coding regions. And that means that this part of the chromosome is actually going to code for a certain protein to be produced. It's going to have a purpose, while this portion of the chromosome isn't actually going to be coding for the production of anything. And we also have to talk about linear versus circular chromosomes, where within eukaryotes and humans we have linear chromosomes. We've got DNA that can be condensed to a linear form, whereas in prokaryotes such as bacteria, chromosomes are going to be circular. There is going to be 1 or 2 strands of DNA that is circular. Last thing we have to do today is discuss plasmids. And a plasmid is a piece of small circular DNA located within prokaryotes that is extra chromosomal, meaning it did not come from the main genetic information of the prokaryote. It is something that came from somewhere else, and it's just a small independent piece of DNA. And it can enter the bacterial or the overall prokaryotic DNA. So here we see our plasmid in this bacteria, and it's just a small circular piece of DNA, but it can integrate itself into the primary chromosome, the primary chromosomal DNA of the bacteria. So there you have it. You got to know DNA and RNA, the nucleotides, the base pairs, single versus double strand, the introns versus exons, and plasmids. We will discuss both of those more in future videos in this unit, but for now, just make sure you have a good understanding of DNA and RNA so we continue talking about them in 6.2.